Good afternoon, everyone. I'd like to thank you for the opportunity to present my work today on epigenetic reprogramming in target tissues by the Histone Methyltransferase MLL. And so I'll just speak briefly to my experience and my pathway and how I got here today. And so I obtained my BS in chemistry at St. Mary's University in San Antonio, Texas. And it was there that I was fortunate enough to be a part of the Mark U-Star program, and I was able to do summer research, and that's really where I became passionate about biomedical research. And so I think that experiences like that are invaluable. I then obtained my PhD at Cornell University in reproductive physiology, where I studied the roles of estrogen receptors in ovarian cancer of the hen. And my interest in steroid hormone receptors then led me to Baylor College of Medicine for my first postdoc, where I examined progesterone receptor signaling and cell signaling pathway crosstalk in breast cancer. And I am currently at the Texas A&M Health Science Center in my second postdoc, where I am looking at how environmental exposures can reprogram the epigenome, leading to increased susceptibility to disease in adulthood. And I'm also writing a KO1 grant application to aid in my transition to independence. And so it is known that the epigenome is plastic, and this plasticity allows the epigenome to be modulated in response to environmental cues so that a developing organism can adapt to any setting that it finds itself in. And so the most famous example of this is the example of a nutrient-poor environment in utero, so maternal starvation. In this case, we can get developmental programming of the epigenome where you get an alteration of the physiological set points in the developing organism so that in a nutrient-poor environment that they may encounter in adulthood, this can confer a survival advantage to the offspring. However, this plasticity is also a vulnerability in the settings in which the fetal environment is disconnected from the adult environment, and then you can get an increased susceptibility to metabolic disease in adulthood. And so we hypothesize that exposure to endocrine disruptors can also disrupt the epigenetic machinery to reprogram the epigenome and alter disease susceptibility in adulthood, including to metabolic disease and cancer. And today I will focus briefly on my studies with the environmental estrogen bisphenol A, or BPA, and its effects in the liver and the prostate. And so this is just a little paradigm that we use, and so we treat an outbred population of rats with orally with low-dose BPA on postnatal days 1, 3, and 5. And then we follow the rats along the life course, and we collect liver tissue at postnatal day 70 and also at postnatal day 240 after feeding a diet high in fat, fructose, and cholesterol. And so we can see in neonatal liver that we have an increase of H3K4 trimethyl, so that's a histone mark, that's an activating mark for gene transcription, at a subset of Hox genes or homeobox genes, which are known targets of the MLL methyltransferase that writes this mark in a subset of these outbred animals. And interestingly, we also see an increase in the H3K4 trimethyl mark at genes related to hepatic fibrosis or steatosis, such as cyclin E1, which you see at the bottom there, and that this reprogramming is concordant with that of the Hox genes. And importantly, this reprogramming persists into adulthood. And this is even in the absence of increased gene transcription, which is very interesting to us. And we find, in fact, that if you feed a diet high in fat, then these reprogrammed genes now have an increased expression. And this is correlated with, we see increased liver weight in these animals. And so this suggested to us that these epigenetic reprogramming can actually prime genes to become responsive to challenge later in life. And in fact, we also see this in the prostate. And so this is a similar treatment paradigm, but in this case, in adulthood at postnatal day 70, we treat with testosterone plus estradiol to promote prostate tumorigenesis. And then we look at prostate lesions. And in the neonatal liver, we see that we get cleavage, which is necessary for the activation of MLL, and with BPA in response to BPA. And then we also see this increase in the H3K4 trimethyl mark at KEGG prostate cancer genes, such as the GREB2 gene. And again, as we saw in the liver, this persists into adulthood, so this reprogramming persists and does not change basal gene expression, but we do see an enhanced response to androgens. And so these KEGG prostate cancer genes are actually now hormone sensitive. And this correlates to an increase in the protein expression of the GREB2, and then we also see an increase in PIN lesions in response to BPA. And so together, the data in the liver and the prostate give us a model where we have environmental exposures activating non-genomic signaling pathways, such as PI3K, AKT, which I did not have a chance to talk about today, which can lead to the activation and cleavage of MLL. And then we can get reprogramming of target genes in the developing tissues, which persist into adulthood and alter disease susceptibility later in life. And so I'm very interested in how this MLL reprogramming all comes about. And so I hypothesize that MLL reprogramming is a mechanism by which the environment can promote the development of prostate cancer. And to study this, I'm going to examine the crosstalk between PI3K AKT signaling and MLL. I'm going to examine how MLL is recruited to reprogram target genes. And I also want to identify an epigenomic signature of prostate tumorigenesis in response to environmental exposure, because no one has really done this before. And I think that it's a very important question. So with that, I'd like to thank you for your attention, and I'm happy to take any questions.
About the chromatin marks themselves, or the recruitment, maybe the differential histone distribution, or what do you think is the mechanism that's happening?
Right, and so we don't know, right? And so this is the first time, usually when you get this H3K4 trimethyl mark, you see an increase in gene activation. And in the prostate, we actually saw that. So there are a subset of genes where the basal expression does go up because the H3K4 trimethyl mark goes up. But what I don't know is if the H3K4 trimethyl mark alone is sufficient, or what's happening with RNA pol 2. So I want to do some grow-seq, and I want to look at other marks, and nucleosome positioning might also be something that is relevant as well.